The following audio recording has been designed for learning purposes only. Do not share, distribute, or provide any type of copies, or this audio lesson will not be made available to you. Good luck with today's lesson.
They want to rush right in, and we get this electric current. And that is exactly what's happening during a muscle cell action potential or a axon action potential. We are opening channels, allowing positive charge to come in, and that positive charge relates to an electric current. And we can measure it. And we know that between the inside and the outside, there's a minus 70 millivolt difference, meaning that it's more negative inside and more positive outside. So how do we maintain this? We've got the sodium-potassium pump. Yes, some sodium leaks in, some potassium leaks out. And these are two positive, right, ions. But the sodium is going to leak in and the potassium is going to leak out by diffusion. So we've got a sodium-potassium pump that pumps 3 sodium out, 2 potassium in. That's a difference in positive charge. 3 in, 2 out. That's why we get other way around. 3 out, 2 in, right? 3 positives out, the 2 positives in, and that helps us to maintain, right? This negative inside. We're pumping out 3 positives. We're only bringing in 2 positives. That helps to maintain this negative situation inside and the positive on the outside. So what kind of channels then do we have in the membranes of muscle cells and axons that are going to allow us to have this electrical current? Okay. Well, we have a couple of different kinds, right? A lot of our sensory neurons and sensory receptors have what we call mechanically gated channels, right? And somehow we apply some pressure, and you know, that's just a drawing, but somebody grabs your arm. You are mechanically opening sensory receptors and neurons to be able to allow positive charge to come in and make it more positive inside. Electrical current. The other kind we have are what we call ligand-gated channels. You know, you've all heard of neurotransmitters. Neurotransmitters get released. They cause something happening, especially in muscle cells we've talked about. Well, those neurotransmitters are what we call ligands. Okay? It's a molecule of some kind. It's not always a neurotransmitter. It might be a hormone, or it might be a growth factor, or it might be some other signal that's being sent to a cell. But in the case of muscle cells and axons, they're ligand-gated. They're going to be neurotransmitters. So the neurotransmitter binds to its spot, and then the channel's going to open. Once again, we're letting in positive charge. That's going to make it more positive inside. Electrical current. Okay. Does that make sense now? Yeah? Okay. Any other questions about it? Good? Okay. All right. The third, and we saw the little video of the binding and the opening. Okay. You can look at that on your own. No. The other kind we have are what we call voltage gating. And they have, in this case, activation gates and deactivation gates. When nothing's happening, we could say at resting membrane potential, it's negative outside, positive outside, negative inside. The channels are not activated. This one is closed. This deactivation one is open though. It's kind of like an extra gate that helps us. When we reach a certain voltage inside, right? So we're going from minus 70 and we're going to go up, up, up towards the positive because we're letting positive charge in, right? They're going to open. So when we get to a certain voltage inside the cell, these gates, the activation gate is going to open and positive ions come through. We're making it more positive inside. At another voltage, the inactivation gate closes. Done. We can't let any more positive charge come in. Okay. So these are going to open and close based on the voltage, the electrical current that comes into an axon or a muscle cell. And depending on what that voltage is, they will either open or they will close. And we figured out exactly what those numbers are when we look at muscle cells and axons. Okay. We know exactly when they're going to open and we know exactly when they're going to close. Okay, so we got 3 different kinds of channels. Now, we have what are called local potentials. The dendrites and the cell body have some channels that open. They're usually either mechanically gated or ligand gated. Okay. And then interspersed in there are a couple of voltage gated. So we said the dendrites are the receiving end for messages. What kind of messages? Neurotransmitter messages. So we're going to have lots of ligand gated channels in the dendrites and the cell body. And when neurotransmitter gets released from another neuron, it's going to bind to those gates and those ligand gates open. And they allow positive charge to come in. So now, in that local area where that ligand-gated channel was, it's now no longer minus 70 millivolts. It's getting a little more positive because we've let positive sodium in. So we say that we are depolarizing the dendrite. So there's our first term, depolarization, making it more positive inside. Now, we get this information coming into the dendrites in the cell body. If it's a lot of information, we're going to open up a lot of these ligand-gated channels. And then we can get an action potential, not just a local potential, but we can get that action potential, that electrical change, right, that current to move all the way down the axon and cause the axon terminals to release neurotransmitter. Yeah. But local potentials have to open up a lot of channels, right? So we say that they're graded, right? If 10 neurotransmitter molecules bind to a ligand-gated channel and open it, are we going to be able to allow lots of positive sodium to go in to make it more positive inside? Not really. That's not a real strong stimulus, right? A stimulus, something that's going to open those channels. It's kind of weak. So the stronger the stimulus, in this case, the more neurotransmitter that's released from the other axon is going to open more and more and more and more of those sodium channels, so we get a bigger stimulus. Okay. They are detrimental. Yeah, once the ligand binds and sodium rushes in, the ligand, the neurotransmitter, releases and the channel closes. So it stops the flow of sodium in. Now, these local potentials on the dendrites opening up those ligand-gated channels can either be excitatory or inhibitory. Some neurotransmitters open channels that make it more positive inside. Excitatory. Some neurotransmitters open channels that allow negative to go in or positive to go out, that would make it more negative inside. That would be inhibitory. We want to be around that minus 70 millivolts all the time in order to get action potentials. These local potentials can start action potentials or they can inhibit them. So it just depends on what neurotransmitter, what kind of channel, whether the channel allows stuff in or stuff out. If it's allowing positive charge to go in, it's excitatory. If it's allowing positive charge to go out, it's going to be inhibitory. Okay? So what we say is, we get a summation. There may be, 30 axon terminals signaling to the dendrites. Some of them may be releasing excitatory neurons. Some might be releasing inhibitory neurons. I said neurons. I'm sorry. Of the 30, some will be releasing excitatory neurotransmitters. Some will be releasing inhibitory neurotransmitters. So it's who's winning. It's almost like a little competition, right? So it's a summation of all of these little local potentials happening at the same time. The more positive it gets inside, the more likely we're going to get a response from that particular neuron. The more negative it gets inside, the less likely we're going to get a response. So it's kind of this summation. I mean, your brain, you have zillions of neurons, right? Axon terminals that are signaling to the neurons in your brain all the time, right? Which ones are kind of winning? Excitatory or inhibitory? Excitatory, inhibitory. So we kind of sum up all of those incoming signals. So that's a local potential on the dendrites and the cell body. If we get enough positive coming in, then we can actually start an action potential down the axon, which will go all the way down that neuron's axon to those axon terminals and cause neurotransmitter to be released from there. So here you go. Mechanically gated, ligand gated. All of these are on the dendrites and the cell body. Makes sense so far, right? We're trying to get that neuron to send a signal, and that signal is going to be through neurotransmitters. But in order to release the neurotransmitter, we have to have this change in current, right? This electrical charge change. So, remember you learned about the axon hillock? Did you learn about that in lab? Right? So, okay, I don't draw very well. Okay. But here's our axon. We've got the dendrites. Here's our axon cell body. Whoops. I gotta go a little wider. Sorry. Erg. Okay. This little area right here. So all these ligand-gated channels are all around the cell body and the dendrites. You have more dendrites sticking out. This is why I don't draw. Looks terrible. But we only have one axon coming off of that cell body. And this little sort of triangular area right here called the axon hillock or the trigger zone. Why? There are tons of voltage-gated channels located there. Huge density of them. Okay? There are a few voltage-gated channels in the cell body membrane, but not that many. And yeah, the book gives you some ideas. Okay, so I mean there are tons of them here. So we've got all of these incoming ligand-gated or mechanically gated signals coming in. And if they're excitatory, they're going to open those sodium channels. Positive sodium starts coming in, and it's coming in, and it's coming in, and it's coming in, and it keeps coming in, and it reaches down here to this axon hillock, and it, boom, it opens up all those voltage-gated channels. There's a minimum amount of change, right? We have to go from minus 70 to minus 55. That's 15 millivolt difference. Right? So all that positive sodium is coming in, coming in, coming in, coming in, making it more positive inside, more positive inside. It's coming down this way, the sodium's coming in. If it gets to minus 55, all of these voltage-gated channels open. And once they open here, they're going to open all the way down the axon, all the way to the axon terminals, where we're going to release more neurotransmitter to the next cell, next muscle, or another neuron. Your brain's doing it right now. That's that time. Colds. That's because spring break is coming. You have to get sick for spring break. Okay? So, we call that minus 55 threshold, right? We're got positive, or we should say excitatory and inhibitory, excitatory, inhibitory. We're releasing all this. We're opening all these ligand-gated channels. If it comes in and it comes up to minus 60, nothing happens here. Nothing happens. But if it gets to minus 55, then the voltage-gated channels in that axon hillock will open and allow more positive sodium to come in. And that starts opening voltage-gated channels all the way down the axon, till we get to the axon terminals where we're going to release the neurotransmitter and signal to the next cell, or the next muscle cell, or the next gland, or whatever that particular neuron is signaling to. Okay? I have this picture. Ligand-gated, they predominate here. There's some mechanical, there's a few voltage-gated, but mostly we have those ligand-gated ones here, because this is where neurotransmitter is going to be released onto all of these dendrites and the cell body. That's the input zone. So, again, a summation of excitatory and inhibitory neurotransmitter being released. If we get a lot of that excitatory, and we're allowing positive sodium to come in, and it's changing that voltage inside, we're going from minus 70. If we get to minus 55, which we call threshold, here at the axon hillock, sodium channels open. And they open all the way down the axon till they get here, where there's going to be more neurotransmitter release to whatever this particular neuron is signaling to. Makes sense? So this is the input zone. Incoming messages. This is, the summation zone, the trigger zone. It's called the axon hillock. And we've got to get those voltage-gated channels in here to open. So we have to let in enough positive sodium to change minus 70 to minus 55. And if we do, then those voltage-gated channels here in this axon hillock are going to open. And once they open, there's a ton of them there. You don't need to know this, but look at the difference. Yeah. 300 times more per micrometer squared. Holy smoke. Tons of sodium come in, positive charge. And that's going to start to spread and open up voltage-gated channels all the way down the axon. till we get to the synaptic knobs or the axon terminals where we're going to release neurotransmitter. This is an action potential. These are local potentials. Okay. If we don't get enough, if we don't get to minus 55, nothing happens. All right. So far, so good. So, yeah. Once we reach that threshold, minus 55, we say the neuron or the axon fires, right? It opens up all those voltage-gated channels. And that's the action potential moving down that axon. Whether it's going this far or that far, the voltage-gated channels are going to open up all the way down the axon until we get to the end and release neurotransmitter. Okay? So, we had that little voltmeter thing, right? And we can get a little drawing, you know, like when you go and you get an EKG and you get a little scribble on a piece of paper. Yeah, we'll talk about EKGs later. We're measuring electricity in the heart. What? Yeah, your heart has electricity too. We see this little spike where all that positive comes in and we see the change in voltage from minus 70 to about plus 35. And then that inactivation gate closes and it doesn't allow any more sodium to come in. And we've measured it with that voltmeter thing and we've been able to take drawings and everything. So that is the depolarization of the axon. And now it's more positive inside and more negative on the outside, on the axon membrane. I pulled this because I thought it was a little bit easier to read. Okay, so I pulled this from another source I have. So, if we use that little voltmeter and those little electrodes, and here we are at resting membrane potential, meaning nothing's happening. Okay, there's no signal. We're going along at minus 70. All of a sudden, we get some channels opening on the dendrites and the cell body. If we get to threshold, minus 55, we are going to open up tons. This is the axon hillock, right? We're waiting to get to minus 55. We're going to open up all those sodium channels all the way down the axon. We call that depolarization. And we made it way more positive inside, obviously, right? 70 plus 35, 105 millivolts more positive on the inside than the outside. Pretty cool, right? And right up here, those sodium channels, that inactivation gate's going to close, and that prevents any more sodium from coming in. Now we have an issue is how do we make it more negative inside again? Well, those sodium-potassium pumps are working, but that's going to take quite a while. And maybe that neuron needs to fire again. Maybe it needs to send another action potential really fast. So, is there something else that we can do to repolarize, bring the inside all the way back down towards minus 70 millivolts, where we're back at resting? There's another positive ion, the potassium, which is K+, that we can let out, right? And then we can pump it back in. 3 sodium out, 2 potassium in. So the sodium-potassium pumps can help us re-regulate where everything is supposed to be. But in the meantime, we want to let that positive out as fast as we can. We can't get the sodium back out that fast because we just let it all in. But there's another positive ion in the inside that we can let out, and that's the potassium. Oh, I know that looks like a minus, but that should be a plus. Sorry. The pluses don't look very good on this picture. So, once the sodium channels are closed, the potassium channels will open. And potassium will start to flood out, we say. Go out, go out, go out. So we're letting a positive charge out, which means we're making it more and more and more and more and more negative inside. We go past threshold, we're heading down towards minus 70, but the potassium channels kind of close slowly. They're not super fast. It's not like they slam shut. So it gets a little extra negative, probably down to about minus 70, maybe down past minus 70 to about minus 80, minus 90, just depends. And so we hyperpolarize it just a little bit. But then sodium potassium pumps are working and we get right back to minus 70. So with those little electrodes, we can get a trace like this on a little machine that shows us what the change in voltage is during an action potential. So we know that we've got to get it to minus 55 at the axon hillock. Right? And if we do, then all the voltage gated channels down the axon will open. And we have depolarized it. We made it more negative inside. I'm sorry. I'm backwards. We made it more positive inside. Right? It goes from negative 70 to plus 30. But again, voltage gated sodium channel, when it reaches plus 30, it closes. Right? That little hangy thing closes and prevents any more sodium from coming in. But we got to get back down to resting membrane potential so the axon can work again. The only way we can do that is by letting something positive out. So at plus 30, the voltage-gated potassium channels are going to open. And they let positive potassium out. And they're open, potassium is flowing out, flowing out, flowing out, flowing out. They start to close right around here, but they close kind of slow, so we get a little extra negative inside for a little while. We call that hyperpolarization. And then the sodium potassium pumps are working, and we get right back to resting membrane potential. This is called repolarization, going back towards minus 70. Right? We're polarized at minus 70. We depolarize to plus 30. We repolarize towards minus 70, and then we little hyperpolarize, make a little extra negative, and then we get back to resting again, minus 70. And this is what we can get a trace while we are looking at what's happening in a neuron when it is being stimulated by some sort of, I don't know what, pain, pressure. Okay, I do apologize. I need to take this phone call. Okay, it's from a doctor's office, so can you give me 1 second? Hello? Hey, yes. I do apologize. You know, when doctors call you in, you try to get them back and you never can. Thank you so much. I appreciate your patience with me. Okay, so this is what the trace is when we are measuring an action potential. And these are the important numbers. Resting, minus 70. Threshold, minus 55. Plus 30. Sodium channel closes, potassium channel opens. We repolarize. They're closing. They close a little bit slowly, so some extra potassium goes out, and that makes it a little extra negative inside, probably minus 80. And then the sodium potassium pumps, they're always working regardless of what's going on here, but they finally can bring us back to that resting membrane potential. Action potential. Okay. So, by the time the voltage peaks at plus 30, the slow opening potassium channels are fully open. They open up really wide, and now we are repolarizing the membrane. We're letting positive out, which is making it more negative inside. They do stay open a lot longer, so we get slightly more potassium leaving, so that makes it a little extra negative inside. So we get a little lower than minus 70, so we hyperpolarize a little bit. And then, whoop, we notice that potassium and sodium literally are switching places during an action potential. The sodium comes in, the potassium goes out. Normally, it's potassium in, sodium out. Okay, so we got that switch. And then those sodium potassium pumps, they never stop. They're constantly working. Because remember, we also have some sodium and potassium leaking in and out just because they can get over the membrane. Okay. And we're back here again. Any questions about what's going on here? Anything not make sense? Have you heard some of this before? Yeah, maybe not. Okay. But this is the electrical current. Right? It's also what, I don't know about you guys, I've been getting shocked by stuff lately.
A lot of static.
A lot of static in the air, yeah. And we conduct electricity this way, but also between person and person. I touched a microscope the other day and it zapped me.
Yeah, I get zapped every time I go to like the grocery store or something.
Yeah?
I go to like reach for like the shelf and I like touch the metal.
The shelf, the metal, yeah. So yeah, we conduct electricity. That's how you get electrocuted. See that? Yeah. Electrical currents. Okay. So on Thursday, we'll do a little something here in class to go over this. Okay? Yes, ma'am.
So you say the negative 30 is the negative 30 repolarizer?
At plus 30,
Oh,
from plus 30 down to a little extra negative, maybe minus 80 millivolts, is the repolarization phase. Okay? So up here at plus 30, okay, all the way down here, and again, we go a little bit lower than minus 70. So we probably go to like minus 80. Okay. So this would be the repolarization. Repolarization, we're going back towards that polarized number. Yeah, minus 70 millivolts is where it's polarized. Okay. When we depolarize it, we get more negative. When we repolarize it, we get more. I did it backwards again. When we depolarize, we get more positive. When we repolarize, we get more negative. And then we hyperpolarize. make it a little extra negative inside, just because those potassium channels close kind of slow. So, depolarize, repolarize, hyperpolarize. And we're using the term polarized because the membrane is polarized to minus 70 millivolts. If something's polarized, that means there's a difference in charge. Negative inside, positive outside. So that's why we say the membrane is polarized. Okay? All right. Good questions. Okay. So here they are. This is kind of cool. I'm not going to take a lot of time to go through this, but there you go. The sodium channel actually has two gates, an activation and an inactivation gate. And the potassium channel only has one gate. But again, depending on voltage. So here you go. In the resting state, we're at minus 70. Right? In the depolarizing phase, we've opened this. Sodium's coming in, it's making it more positive, more positive, more positive. This one says plus 30, plus 35, it's all the same, okay? Making it, or depolarizing, making it more positive inside. Then all of a sudden, these gates close, this gate opens, and now we are repolarizing, bringing it back down, back down, back down, back down. This gate is closing, but it's closing a little slowly, right? So we get to minus 70, and we're going to go just a little further. Oh, it didn't go further here. Okay. We go just a little bit further than minus 70, and we hyperpolarize a little bit. But that's showing you the condition or the situation with those gates. Okay? And then the gates, we say reset, and they're ready for another action potential.
When the cell membrane is at its resting membrane potential, the activation gates of the voltage-gated sodium ion channels are closed, and the inactivation gates are open. Voltage-gated potassium ion channels are closed. Depolarization is initiated by a stimulus which makes the membrane potential more positive, causing the voltage-gated sodium ion channels to start to open. As threshold is reached, many sodium channels open. Sodium ions diffuse across the membrane, causing depolarization. Voltage-gated potassium ion channels also begin to open, but more slowly. Therefore, depolarization occurs because more sodium ions diffuse into the cell than potassium ions diffuse out of it. As the membrane potential approaches maximum depolarization, the inactivation gates of the voltage-gated sodium ion channels begin to close, and the diffusion of sodium ions decreases. The potassium ion channels remain open, and potassium ions continue to diffuse out of the cell. The increased potassium ion permeability lasts slightly longer than the time required to bring the membrane potential back to its resting level. The extra efflux of potassium ions causes the membrane potential to become slightly more negative than the resting value. After the voltage-gated potassium ion channels close, the active transport of sodium and potassium ions reestablishes the resting membrane potential.
Now, this is fast, right? Some of these neurons are signaling at 300 miles per hour. Well, 270, I just rounded up, right? Yeah,
More optimistic.
that's fast. This is happening so fast.
When the cell
No, thank you. Okay. All right, so when this is happening, and even though it's happening so fast, we have what we call refractory periods. This is a period where it's kind of resistant to stimulation, right? Yeah. We have the absolute refractory period. Hey, if we already opened all those sodium channels, we can't open them anymore. They're already open. We call that the absolute refractory period. So we could keep giving the neuron a stimulus, but they're already open. You can't open them anymore. There's nothing else to open. So it doesn't matter how big the stimulus is, we're not going to get a bigger action potential. They're open. So we call that the absolute refractory period. Okay, but now, so here we are, threshold, right? Here's threshold. That's where we have to reach. We've depolarized. We're repolarizing. Oh. We started down here at minus 70, and that was a 15 millivolt difference. Now we're coming down. This was plus 35. Now we're starting to come down. So, like right here, this is the absolute. They're already open. You can't do anything else. They're open. But now it's starting to get more negative inside. Okay. Plus 25, plus 15. Oop, I don't. Plus 5, plus, well, there's no plus, but 0. Right? You get the idea. Well, those gates are closed. We're coming down, we're coming down. What about if we gave it a stimulus that would get it up to 0? Suppose we gave it a minus 70 millivolt stimulus. Could we get it from minus 70 to 0 and get those to open again? Yes, we could. Because they're closed. The sodium gates are closed at plus 35. Right? And they're resetting, their activation and deactivation, they're changing, so it's getting ready. We're making it more negative inside, but we have to give a bigger stimulus. We have to give more than 15 millivolts to get them to open again, depending on where we are in the repolarization. So this is the relative. God, what is wrong with this thing? It's me, it's not it. The relative. It's always me. Just take my word for it. It's the relative refractory period. And how do I remember that? I goes, we have to give it a relatively larger stimulus to reopen those channels. So we need a really strong stimulus to match wherever we are in this repolarization phase. If we're at minus 40, we'd have to go from 70 to 40, we'd have to give it 30 millivolt stimulus. If we're at 0, we have to give it a 70 millivolt stimulus to get them to open again. So it just depends on where we are in this repolarization phase as to how big a stimulus we need to get those sodium channels to open again. So, absolute, they're open. We can't do anything else. No matter, we could give it 100 gigajoules. I don't know, is that even a thing? Right? We could give it as much stimulus as we want. But the sodium channels are already open. They can't open anymore. They're open. So we call that absolute refractory period because it doesn't matter what the stimulus is. But as we are repolarizing, a bigger stimulus could get them to open again. Okay? So we call this the relative refractory period. Right? I mean, this stuff is happening in milliseconds. Right? So even though you're thinking, oh, we got to wait, no, we're not waiting a long time. It happens really fast. I'm going to go back here. Oh, they don't have the time on that. Sorry.
What?
Shut up.
So I have a question.
Yeah.
So like when like a doctor does defibrillation, and like, is that because they're trying to get that
We're
to
going to
open up again, or is that something completely different? Because they're giving that stimulation and where they increase the voltage.
your heart is influenced by your brain, but it has its own electrical system. You have some specialized cells in your heart that start these action potentials, and when it starts, the first signal causes the atria to pump, the second signal causes the ventricles to pump. So it's sending signals to the heart muscle cells. Okay. What happens is sometimes that electrical system gets haywire. That's a good word, right? It's not normal. And if you go into fibrillation, that means that the atria are not working together, they're just doing this, or the ventricles are just doing that. So it's not effectively receiving and pumping blood out of the heart. So the electrical defibrillators are to shock the heart with the electrical system to try to get it back into its normal rhythm. Mm-hmm. Very important. We're going to talk about that when we get to the heart. All right, so here you go. Here's a nicer picture than what I drew.
Where's this one?
There you are. Right. Absolute refractory period. All right, they've gone a little further, okay, over. It is actually going to be a little bit more here as I was talking, right, as we go. As those channels are resetting, a bigger stimulus will allow us to jumpstart it again. Okay. I should have taken that one in. I don't like it. But it's in your book, so I wanted to explain it. It's a little bit over more. Okay. Okay. And again, you know, the more that we are going towards the negative, the more those sodium channels are resetting. Okay. Makes sense? Yes? Questions? All right. Unmyelinated and myelinated fibers. Yeah. If we don't have myelin around the fibers, and the fibers are the axons, that means we literally have to open every single sodium channel all the way down the axon. It's a chain reaction. You know, once the axon hillock fires, there's enough sodium coming in, those channels open, there's enough sodium coming in that it keeps moving and then opening more and opening more. It brings all of those to threshold, the minus 55, and we just keep opening and opening more and more and more and more and more voltage-gated channels down the axon. If there's no myelin, we have to continuously open each and every single one. So we call it continuous conduction. It's like a chain reaction. Once the axon hillock lets in that sodium, it's going to open up every single voltage-gated channel all the way down the axon to the axon terminals will release neurotransmitter. That's good for short distances. Don't necessarily need myelin. But what about if you're going from the tip of your big toe all the way up to your spinal cord? Well, that's a bigger distance. And that's where the myelin comes in. Okay? So here you go, unmyelinated fiber. We've got the axon hillock to fire. We are opening up every single one of these. Right? And behind it are the refractory periods. It's getting reset so another stimulus can cause another action potential. But there you go. All the way down, all the way down, all the way down. And it's going to move like a chain reaction all the way down until it gets to the axon terminals and we release neurotransmitter. Yes. Saltatory conduction. You know, especially in the nodes of Ranvier, yeah, there are tons of these voltage-gated ion channels. Underneath the myelin, really that myelin is wrapped super tight around that section of the axon. So nothing's going to really get under there. There might be a few sodium channels, but really it's in those little gaps in between. The nodes of Ranvier, or what are they calling them now in your textbook? Myelin sheath gaps. They don't want to name it after the guy who figured this out. There's tons of these here. And actually, when you open the sodium channels here, enough sodium goes underneath the myelin to open up the voltage-gated channels in the gap. And then enough sodium comes in at that gap, goes underneath the myelin and opens up all the voltage-gated sodium channels in the next gap. And it follows all the way down the axon. So this is what we call saltatory conduction. It's kind of like jumping from node to node to node. Like this. So again, we've already activated the axon hillock. Sodium is coming in. There's enough sodium coming in here to open up all the voltage-gated channels in the little gap. Sodium rushes in, and it's enough to move all the way along to the next gap and open up the sodium channels there. Sodium rushes in, and again it spreads to the next gap, and the next gap, and the next gap. So it goes a lot faster. And that's how you know you stubbed your toe almost immediately. So for those really long, right, 30 centimeters, that's about that long. That's a long way to go. And this kind of conduction makes it go super fast. So underneath here, yeah, that myelin is wrapped so tight that nothing really gets under there. But in those gaps, we're loaded with those sodium channels. So, there you go. We're going to jump from node to node to node to node, all the way down. And then behind it, it's resetting. It's going through repolarization, hyperpolarization, getting ready to be able to respond again. I like this picture. This is cool. Look, the difference between continuous and saltatory conduction. Look at 1 second. They both start at the same place, the axon hillock. After 5 seconds, it's a little further. After 10 seconds, it's almost at the end. And here it's still coming down. Because it has to open up every single one of those sodium channels. Super fast, super slow. But again, you know, the neurons in your brain some of them are traveling all the way out, down your spinal cord and all that. But the ones that are within the cerebral cortex where they're signaling to each other, they don't have that far to go. Some of them are real short like this. They don't need myelin. So it makes it faster. I mean, in 10 seconds, it's still not a big deal, right? But if you're going from your big toe all the way to your spinal cord and then up to your brain, that's a long way to go. All right, there is one other thing I want to mention. I don't know if your book does this or not, but we refer to it as the all or nothing principle, okay? There aren't any big or small action potentials. Okay, they're all one size. Every time it's going to go to plus 30. The next action potential plus 30. The next gap plus 30. The next gap plus 30. It's always the same. When the action potential occurs and the membrane reaches threshold, it's going to stay at plus 30 all the way down. If we don't reach minus 55, we could be at minus 56, nothing. We got to be at minus 55. We either get there or we get nothing. And if we get it, it's always going to go plus 30, all the way down the axon. It's not going to go plus 30, plus 32, plus 30, plus 29, plus 25. It's never going to diminish. It's always going to go up to that plus 35 or plus 30, whichever we got. Okay. So we call it the all or nothing principle. Okay. All right, synapses. So we've got the action potential. We're down now at those axon terminals. I mean, this kind of makes sense. You've got the synaptic knobs, the axon terminal of one neuron signaling to the cell body or the dendrites of a second neuron. And where they're going to signal is called a synapse. And remember, there's like a little cleft in between. They don't actually touch each other because they're releasing neurotransmitter. And we have this little gap, we call it a cleft, in between them. Well, the one that's releasing the neurotransmitter is called the presynaptic neuron. The one receiving the neurotransmitter, or responding to it, is the postsynaptic neuron. And then that little cleft is the synaptic cleft. So this is kind of like, I guess we might call it a functional connection. It's not really an actual touching connection. It's not a physical connection. But it's more like a functional connection because it's going to allow the next cell, whether it's a neuron, a muscle, a gland, to respond to the neurotransmitter. So where they're going to meet, this little spot, and in the muscle cell we called it the neuromuscular junction, right? The motor end plate spots on the muscle cell where the receptors were for the neurotransmitter. The axon terminal came down, released it, it went across this little cleft, and the muscle cell responded. Presynaptic, postsynaptic. A synapse is this kind of functional junction. I don't know how else to explain it. Does that make sense to everybody? No? How come? I can draw again. You'll be sorry. Okay. Right. So, this is the axon terminal, right? And we've got all the little vesicles with the neurotransmitter in here. And then we have our cell body or our dendrites. And in this case, we have a dendrite. I'm not very good at drawing, so bear with me. Those are my dendrites. Looks like a rabbit. And this is the synapse. It's almost touching, but not really. And then these little vesicles are going to come up, and they're going to merge with the membrane, and they're going to release neurotransmitter, which will then bind to those ligand-gated channels. Okay. That little tiny space in between here is the cleft. You know somebody who has a cleft chin? Yeah, it's that little space in between the two parts of their chin. Yeah. So it's a little cleft, a little space. Okay. So if this is a synapse, this is the presynaptic neuron, and this would be the postsynaptic neuron. Pre means before, post means after. And this meeting where these two come together is the synapse. Okay? Yeah, you gotta ask. Okay. I'm pretty sure I have a picture, but I like drawing rabbits. There it is. There's my picture. So here you go. They don't touch. It's a functional junction, not a physical junction. So this would be presynaptic, before the synapse, and this one is postsynaptic, after the synapse. The little space in between is the synaptic cleft. That's where the neurotransmitter gets released, comes over, binds to those ligand-gated channels, and boom, we're going to get some opening of those sodium channels. Better than my picture. Although mine looks cute. Should we give him some, no. Whiskers. Okay. We're going to talk more about these electrical synapses when we talk about the heart. We just mentioned that your heart's got an electrical system. Okay? And your heart, your atria and your ventricles have to pump in unison 2 atria, 2 ventricles, 2 atria, 2 ventricles. Because if they don't, if they're just pumping all over the place, then we're not very efficient at getting blood where it needs to go. So those cardiac cells are connected with those little gap junctions, which are like little tunnels, and they're literally going to be able to send an action potential from cell to cell to cell to cell. And we'll talk more about them when we get to the heart. The ones that we have here, right, are our chemical synapses, because we're going to release neurotransmitter. So a chemical, right? It's going to bind to a receptor, a ligand-gated channel on the other neuron, the postsynaptic neuron, and open a channel there. And remember, these can be inhibitory or excitatory. And we're going to sum up. What are we receiving? Are we receiving more excitatory or more inhibitory? Are we going to have an action potential? Are we going to reach threshold or not? So there's 2 different types of synapses. And again, the synapse is a junction, but it's a functional, not a physical connection. Okay, there you go. This would be an electrical synapse, and this is what happens in your heart cells, right? Yeah. We're just going to be sending stuff back and forth. And then your chemical. And again, this is your, a neuron, right? This is the cell body or a dendrite of another neuron, and there you go, we're releasing it, it's going to bind, and it's going to cause something to occur in the next cell. So we'll go back to those electrical ones when we get to the heart, because that's where we're really going to see them a lot. All right, so the synaptic knob, yeah, that's at the end of the axon terminal. That's that little round bulb. Okay, so they call it a knob, because it does kind of look like a doorknob, doesn't it? That's what it reminds me of, like a round doorknob. Okay, so we call it the synaptic knob. It's going to have all those vesicles in there. It stores them until it's signaled to release them. And then it releases it into that synaptic cleft. And then the neurotransmitter will bind to the ligand-gated channels, and hopefully we get an action potential. There's a couple of different ways that axons can signal to another neuron. They can signal on the dendrites, they can signal on the cell body, or they can actually sometimes signal right onto the axon. So we have a couple of more different types of these chemical synapses. Axodendritic, axon to dendrite, axosomatic. Remember, the cell body is also called the soma, axon to soma, cell body. Or axonic, axon to axon. That's what these words are saying. So, axodendritic, axon to dendrite. Axosomatic, axon to soma. Axon, axonic, axonic, axon to axon. So it just depends on where we are. When the brain, we're out in the peripheral nervous system, wherever we are, these are the different ways that they can signal. And here you go. These are what these little synaptic knobs look like. And look at this. It's inundated with zillions of them. And again, these signals, are they inhibitory and excitatory, inhibitory, excitatory, a mixture? Are they all excitatory? Are they all inhibitory? Scanning electron microscope picture, isn't that cool? I wonder who they got it out of. I was just reading something on the internet where they found 100,000 million year old insect wing fossil. That means they were here long before us. Oh, scary. No wonder there's so many of them. But these are cool, right? Yeah, they showed a picture of it. Did I take you off track there? Isn't that kind of cool? I don't know, I just wanted to make sure you're still paying attention. Pretty cool. All right, and there you go. Axonic. Look at it, right to the axon hillock. Axosomatic, right to the cell body. Axodendritic, onto the dendrites. Just where's it signaling to? So we can have synapses all over the place. Just another little picture. Okay. I thought this one was better, but this one's from your book. They made it, again, axon to the cell body, axosomatic, axodendritic, where is it? Dendrite, somatic, axonic. So, I thought the other one was a little prettier. Okay. Kind of gave you an idea of where everything is, too, rather than just those round drawings. Okay, so the presynaptic neuron contains the synaptic vesicles with the neurotransmitter. How do they get released? By exocytosis, which means that these come up, they merge with the membrane, and then they release their neurotransmitter. Okay. The postsynaptic neuron is going to have usually ligand-gated ion channels. Now, if it was signaling to a gland, there might be a different kind of receptor, but we're not really talking about the gland right now, so it's not a big deal. But think of it as a ligand-gated ion channel, okay? Open it, allow positive in, change the voltage, start an electrical current. We came back to this again. So here we are. Signaling. Merging with the membrane, releasing the neurotransmitter. And then we have a little picture. Now, remember in muscles where calcium was really important? Guess what? We need calcium here too.
Action potentials arriving at the presynaptic terminal cause voltage-gated calcium ion channels to open. Calcium ions diffuse into the cell and cause synaptic vesicles to release acetylcholine, a neurotransmitter molecule. Acetylcholine molecules diffuse from the presynaptic terminal across the synaptic cleft and bind to their receptor sites on the ligand-gated sodium ion channels. This causes the ligand-gated sodium ion channels to open, and sodium ions diffuse into the cell, making the membrane potential more positive. If the membrane potential reaches threshold level, an action potential will be produced.
Okay? Synapses. Pre and post, before and after the synapse. Calcium. This just walks us through what's happening. Calcium again. Right? That's why we need to maintain calcium levels in the blood. We need it for muscles. We need it for bones. We need it for our neurons to work. Right? Especially the synaptic knobs to release the neurotransmitter. So make sure you eat your vegetables with calcium in it. You need lots of calcium. Okay? It's going. The action potential causes those calcium ion channels to open. Calcium diffuses in. There's a little pathway that occurs when the calcium will trigger that exocytosis of the neurotransmitter. The neurotransmitter then diffuses across that tiny synaptic cleft. It binds to its receptor on those channels and causes them to open. The opening of the channels, it's either an excitatory or an inhibitory neurotransmitter. So we get what are called excitatory postsynaptic potentials. There's a potential to get an action potential in, yes, say it right or wrong? All right, remember we said that some neurotransmitters are excitatory, some are inhibitory. So if it's an excitatory neurotransmitter that gets released, it's going to be an excitatory postsynaptic potential. The potential is possibly getting enough excitatory neurotransmitter release to cause an action potential in the next neuron, the postsynaptic neuron. So that's why we call it excitatory postsynaptic potential. It's an excitatory neuron. It's going to the postsynaptic cell, and the hope is, the potential is, to get it to have an action potential. I kind of wanted to explain the way they call it this. Excitatory neurotransmitter, postsynaptic, signaling to the postsynaptic neuron, we'll call it a neuron in this case. The potential is we're hoping to cause an action potential in the next neuron. Okay? So excitatory postsynaptic potential. If it's an inhibitory neurotransmitter, then it would be an inhibitory postsynaptic potential. We don't want an action potential to occur in the next neuron. Now, if we leave neurotransmitter out there in the synaptic cleft, it's going to keep signaling. So, it either is quickly transported back into the synaptic knob, we call it reuptake. It can be metabolized into inactive compounds by enzymes, meaning we break it down so it's no longer active. Or it can diffuse away from the synaptic cleft, and some of the neuroglial cells, right, the astrocytes or the satellite cells, can take up some of that neurotransmitter. That's especially true in the brain with those astrocytes. Okay. Calcium, and I have a picture to go along with the little video. Okay. Calcium comes in, there's a cascade of events that cause these vesicles to come up, merge, release, bind to their ligand-gated. So now we have a postsynaptic potential. Hopefully we're going to get a nerve impulse or an action potential in the next cell. Sorry, my turn. It's contagious. So these neurotransmitters can be excitatory or inhibitory. Neurotransmitters, there are over 100 compounds. I'm not going through these, we're going to skip over them because I'm not testing you on them. But if you are interested, you can either read about them in your book or you can read this. Right? But again, they are made in the cell body. They are transported down by anterograde movement down with the motor neurons down to the axon terminals, where they are stored until they are being released. Okay? Calcium causes the vesicles to merge, and then we get, boom, action potential, hopefully. Okay. So again, excitatory neurotransmitters or inhibitory. And you can go through these on your own. I'm not going to go through them, right? But acetylcholine is one of the most common. We use it for muscles. Monoamines, epinephrine, norepinephrine, you may have heard of some of those, okay? Amino acids on their own. The amino acids make the proteins. These on their own can do it. And what we call neuropeptides, okay? They're short chains of amino acids. They're not really like a protein size. They're just like really tiny. Okay. So there's some pictures of what they look like. In your, I don't know if you have this one in your book. I believe you do. Okay. So if you want to read about neurotransmitters, I'm going to let you do that on your own. Okay. But once again, Neural integration occurs because, there are lots of synapses. There are lots. We saw that one cell with all those little synaptic knobs, right? The axon terminals. I mean, it's all there. Okay? So, all of that has to be passed from one place to the next place, to the next place, to the next place. Sometimes you've got one neuron signaling to another neuron, to another neuron, to another neuron, and then it gets to your brain. Okay? So we sort of have these pathways that we have to send that information along. Okay. So why do we have all these synapses? Believe it or not, they can process information, they can store it, they can make decisions. Chemical synapses are the decision-making, your book calls them devices of the nervous system. The more synapses a neuron has, the greater its information processing capabilities. I mean, it always amazes me. You've got these little funky looking cells signaling with neurotransmitters, and yet they're storing stuff. They're allowing you to remember things. How to get home, what your name is. In my case. All right. Process information, make a decision. What am I going to have for lunch today?
That's a good question.
Where am I going to go after work?
Crazy.
Well, that's a given. We all go crazy after we get home. But, what am I going to do? Should I buy this? Should I not buy it?
Buy all the bread and milk.
Tonight, before it snows tomorrow. It also is, who are you? What's your personality? Are you nice? Are you mean? These little neurons and these neurotransmitters do all that. I don't know, I kind of think it's
That's cool.
fascinating, right? We don't even know half of what we should know about the brain. We don't. Okay. So, neural integration then is the ability of those neurons in your brain to process information, store it, recall it, make this, you name it.
So question.
I often feel like mine aren't firing when I'm like, I don't know what I want for dinner. Or, I don't know where I'm going.
Oh, mine
I don't know what I'm
completely
doing.
die when it comes to food.
Okay.
Just like stop working altogether.
I feel like they're not communicating properly.
So like for the movie, I think it's called Lucy.
Yeah.
And she like had this drug that just like made her with something like.
No, never. Not gonna happen.
I'm just saying, like, could like with all that firing, would it like, I don't know.
I think it would just blow her head off. I mean, she's not going to become part of the computer system and part of the universe. I mean, but I think it was far-fetched. It was really good science fiction, but I just don't think that can happen. In the future, can we have drugs that will enhance our ability to process, store, recall, all that? I'm sure there will be. Yeah. As we learn more and more about these neurotransmitters, how they affect the brain, what parts of the brain, people who are deficient in certain kinds of neurotransmitters have,
Well,
you know,
pseudopoly this for Alzheimer's, isn't it?
well, I don't think so. Well, I think it's the dopamine, and that's also for Parkinson's. So there are a lot of things we just don't know. We don't know how those brain cells are reacting. We don't know what's going on there. So there's still a lot to learn. Oh yes, in your lifetimes, there may be a pill you take that'll just cure Alzheimer's, or cure Parkinson, cure dementia, cure whatever. There may be in your lifetime. We just don't know. There's a lot we don't know. And they're working on it. All right, so again, excitatory postsynaptic potentials are going to cause more sodium to flow in, hence we're going to have something happen in the next cell. Okay? Inhibitory usually hyperpolarize it. It usually opens something that makes it more negative inside. It may, in this case, we've talked about potassium channels. It may open a potassium channel and potassium floods out. That's going to make it more negative inside. That's going to make it harder to get to minus 55. If we're at minus 90 or minus 100, we have a long, big stimulus, right? So sometimes we want the neurons to fire, sometimes we want them not to fire. So it just depends. So all of these signals are being sent to the neuron. Okay? And here, an EPSP, excitatory postsynaptic potential, would make it more positive inside. An inhibitory, IPSP, inhibitory postsynaptic potential, would make it more negative inside. The likelihood of an action potential occurring here is going to be less because we need such a bigger stimulus. Here might be more likely. We're getting closer to that minus 55. But we do get what we call summation. I mentioned this here, where we're getting all these different signals coming in, and the axon hillock literally is kind of summing up all that's going on, right? So one neuron can receive input from thousands of other neurons, and that's what makes the brain so amazing, right? Some are going to be excitatory, some are going to be inhibitory, and that's where we get that summation. It's the net effect. Are we getting more inhibitory, or are we getting more stimulatory? Okay? And again, minus 55. And once we hit that trigger zone, it's either going to happen or it won't, right? And so the balance between getting all these signals is what's going to allow the nervous system to make a decision. Do I need to do something or not? So there you go. Here's a nice little picture. This comes from another source. I thought it was cute. So this is just 5 different neurons signaling to one. Yeah. Some of them are excitatory, some of them are inhibitory. Here we have 3 EPSPs, excitatory. There's only 2. There's a probability that this will get excited. If it was reversed, it probably wouldn't get excited. Wouldn't be able to get to the trigger zone and cause an action potential. So it just depends. But they're being signaled all the time. And this summation can happen in 2 different ways. Temporal, time, or spatial, number. Right? If we get lots of stimulus in a short period of time, there's likely that we're going to get action potentials. If we get lots of different neurons signaling to one, we're probably going to get an action potential. Right? So I got a little picture for this. So here you go. In spatial summation, right? 2 signaling to 1 is better than just 1 because now you're getting lots of neurotransmitter, likelihood that you're going to get to threshold really good because 2 neurons are signaling. Over here, and with 1, if it keeps signaling and signaling and signaling and signaling and signaling and signaling, it finally gets us up to threshold, and then we get an action potential in the next neuron. Okay? So spatial means many signaling, and temporal means time, like the tempo in music, right? So continuous signaling to it is likely going to get this one to threshold. So we sum up all of those signals. Memory! We should have talked about this a long time ago, huh? Yeah, all right. Memories are stored by facilitating or inhibiting synaptic transmission, right? How many times have you forgotten something? Yeah, it gets worse as you get older. But short-term memories, these can last for seconds or hours. But it really is limited to 7 or 8 pieces of information. Remember how I told you study for 30 minutes to an hour a day? Otherwise, you're going to forget everything? Yeah, it's true. If you stop reciting it, if you're distracted, or now you got to learn something new, you're going to forget whatever it was. Right? Repetition, 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 repetition. Long-term memories, when we convert those short-term memories to long-term memories, yeah, they can last a lifetime. And as far as we know, we have a limitless capacity to store memories. It's just, are we storing them? However, and we're not going to go into any detail, but it does require some structural changes at the synapse, right? For example, some real brief things, more vesicles, more vesicle release sites, more presynaptic axon terminals, more postsynaptic dendrites, So there's a little remodeling that goes on in those synapses. We're not making anything new, but we can add things there. Transfers from short-term to long-term memory. This is the one we have the biggest problem with. Emotional state. Believe it or not, we learn best when we are alert, motivated, and aroused, not distracted. Rehearsal, repeating, rehearsing, enhances the memory of those things. I used to read out loud. I know that sounds funny, but I was hearing it again, I was reading it, and I was saying the words. All of those things were a form of rehearsing what I was trying to learn. How do you learn a song? How do you learn a new song that you like? Keep hearing it, keep singing it, right? Rehearsing it. Association, trying to associate new information with old information you've already got. Okay? I just said temporal, like the tempo in music. Okay? So trying to associate those words with something you already know. That's going to help you to transfer it into long-term memory. And one of the ways you do this is when you're sleeping. You gotta get sleep. And I know that's tough for students because you're always out partying and then at the last minute you're trying to study. Kidding. But you do need to sleep. You need to be rested. That's the only way where you can be alert, motivated, and aroused. If you're sleepy and you're trying to study, it's not going to work. All right, then we've got all kinds of memories. You can read through these if you're, there was just some interesting stuff. You can read through that if you're interested. Okay? So, any questions? I should do short-term memory the first day, right? So, as you all are aware now, we are postponing that exam that we were going to have.
Good luck with today's lesson.